Hello everyone, let's start the next class of cell signaling course. If you recollect, in our previous class we understood how phosphorylation of receptor can drive signaling in different ways by recruiting adapter molecule or activating next set of proteins, which could be second messenger or a kinase, which perpetuates the signaling further. You have been hearing a lot about the molecule which is kinase, and by now it must be by heart to you that it utilizes ATP and breaks it down to ADP and inorganic phosphate, and this inorganic phosphate is transferred to a suitable acceptor molecule. How do we know that your kinase is working? So in today's class we will see how we can assess the activity of a kinase, and then we will get into different types of receptor. We'll see then which way they are present in different living systems, and then eventually we'll try to touch upon the signaling system which is dependent on receptor and it is abundant in the microbial world. So as I just mentioned, your kinases, the most abundant type of molecule in this case, which is related to the signaling. They could be soluble, they could be membrane bound. I have been emphasizing it multiple times so that you just understand that how important these molecules are. And also they have these molecules have a domain which exhibits specific kinase activity. I hope you all understand that domain is a part of protein which can independently fold and retain the activity which it is supposed to have. In case of kinase, the domain when isolated should fold properly and exhibit the kinase activity indistinguishable from the complete protein. So, and specific kinase activity would mean that whether it is against serine, threonine or tyrosine, or for that matter any other residue which we have spoken, whether it is histidine or sugar or lipids. So what generally these kinases do? They use ATP and transfer the phosphate moiety to the target amino acid if we talk about the target to be a protein which is receptor here. So think about a scenario where you have this amino acid with OH group, because all three residues have this OH group. Even when you go to the lipid moieties or when you will go to the nucleic acid, you will find, or sugars, you will find this OH group is very amenable for transfer of the phosphate residue. So when you have put together the amino acid with OH group and the ATP, it can take two paths to come to this final product where amino acid is transferred with this phosphate and ADP is generated. Normally you can think of that there is an adduct which is forming between the ATP and the amino acid, or you can think of that first ADP starts to hydrolyze and the inorganic phosphate comes much closer to the amino acid, eventually leading to the formation of this particular product. And we all appreciate by now that this phosphorylated version of the target moiety is very important. So how we can assess the kinase activity? Not just kinase activity, sometimes we need to assess whether the ATP is binding or not, or GTP is binding or not. We will not just talk about kinase and ATP binding and hydrolysis. We can also consider this explanation which we are going to see for GTP as well, because I am sure you recollect that how important are the GTPases or the G proteins and their classification that they bind to GTP and hydrolyze GTP. So not only that you need to understand the kinase property and release of phosphate group, you also need to understand if the binding of ADP or ATP is happening to the kinase. Also remember that this is first step towards the hydrolysis of ATP into the ADP and phosphate. So whether it is ADP or whether it is ATP or GTP, we know that they carry the three phosphate groups. The base changes, in case of ATP it is adenosine, and for GTP it is guanosine. So whether it is ATP or GTP, you will have 3 phosphate groups, and from proximity to the adenosine moiety, they are called alpha, beta and gamma phosphate. Now, when ATP is hydrolyzed, in one of the earlier slide you saw that ATP is basically was like ADP and phosphate. So, this third phosphate group is what is hydrolyzed. So, if you want to understand the hydrolysis which is mediated by the kinase, you can use an ATP analog where this gamma phosphate is labeled. There was a time when a lot of radioactivity dependent assays were performed, where this gamma phosphate is replaced with the radioactive phosphate, which is P32. So, when in experiment you have ATP where gamma phosphate is labeled and you provide a kinase, what will happen? The kinase, by virtue of its activity, will cut this bond and release the radioactive PI, and the radioactivity of released material or free material can be detected using the scintillator counter. But when you want to study the binding aspect of this, you cannot afford to have the gamma phosphate labeled, because this will be eventually cleaved by the kinase. So, in that case, you use an analog of ATP for which you have beta phosphate which is radio labeled. So, what will happen, that kinase will bind to this ATP, it will cut the gamma phosphate, but your kinase will be now in adduct with ADP which has the radio label. So, by utilizing different types of versions of ATP, one can assess the hydrolysis aspect mediated by kinase, or the binding property which is a prerequisite for hydrolysis event. And this will go same whether it is ATP or GTP. Just what is going to change, that in case of ATP you would prefer to use kinase, and for GTP you will prefer to use GTPase. But nowadays radioactive nuclei or radioactivity is not preferred. There are better versions which are fluorescence dependent. Which are not harmful. So, there are ways where people have generated fluorescent version of these ATP and GTP analog. So, you can notice here is that this represents the nucleotide analog, whether the 2' or 3' is where the label is present in terms of fluorescent moiety. So, this is what is going to fluoresce. So, now you can think of same experiment, you want to understand the phosphorus. Now, you can understand what is the beauty of this thing is that it is independent of whether phosphorylation, it is independent of whether it is a hydrolysis event or the binding event, because the fluorescence which comes from here, it will always remain bound to the protein. So, in this particular case, if you are using MANT-GTP, you will realize that once this molecule has bound to the ATPase or the kinase, it will hydrolyze this molecule, and you will see how much of the signal is being produced over there. How with time you will see more hydrolysis happening and more of the signal is emanating. Basically, you can record the fluorescence of this molecule which has the maximum wavelength, emission wavelength of 448 nanometer, right. So, utilizing a better variant in terms of harm which it can create, right. We saw the radioactive nuclei creates a problem. So, here you can have MANT-ATP, you can have MANT-GTP, right. But if you notice here, you would realize that it will be better, this particular kind of setup which is shown here, it will better give you response for binding experiment, not the hydrolysis experiment. Why? Because here the phosphate residue which will be released and as a consequence of kinase activity or GTPase activity will be lost, and you will always notice what is binding to the kinase or GTPase. So what people have done, how people have underscore some of these activity which is mediated by kinase or by GTPase. These are old experiments as well as fluorescent analogs are used, but idea remains same, how you will assess this. So think about a situation where you start an experiment with the GTPase. Now this GTPase, when you have set the reaction and allow it to incubate for some time, now you add GTPase. What GTPase will do? It will hydrolyze GTP. What you are measuring here is relative fluorescence intensity. Okay, so you will notice that the signal for fluorescence goes up, which means that GTPase is working fine and more phosphate group is being released. Now think about a scenario where GTPase was working perfectly fine because you had something like MANT GTP, which was fluorescent analog. At some point of time when the reaction is nearing its peak, you add GTP, which is non-fluorescent. What it will do? It will compete with the fluorescent MANT GTP analog, and thus you will see the fluorescence going down because more and more GTP will be occupying the space where earlier MANT GT was lost. So basically you will have assessment of the GTPase activity, or for that matter kinase activity, by utilizing these fluorescent version of this. Further, more detailed studies can be performed where you will notice that you can set up the reaction. Again, this is an example for GTP binding and GTP hydrolysis which we saw in the previous case. It can be similarly done for the kinase activity and so on. So what is done here is that you start the reaction by having the GTPase and the MANT GTP analog and notice the fluorescence intensity. So with time you will see more activity and it reaches to the peak of fluorescence. If you think about the binding, so what you are realizing that with time it's completely reaching the saturation. All the GTPase has bound to this molecule. Now what you are doing is that you are adding different concentrations of the unlabeled molecule. In case of no unlabeled molecule, you will notice that this reaction peaks to its maximum value. But when you increase the presence of unlabeled GTP or ATP with respective reaction, you will see suddenly that it cannot achieve the peak. Why you feel that this is not achieving the peak? Because you are measuring the fluorescence intensity. If you are putting unlabeled molecule more and more, you are affecting the fluorescence which can be obtained from this. So basically you have different ways to measure the ATPase or kinase activity. Similarly, you have similar ways to measure and assess the GTPase activity. Now let's see how we move and make sure that these things are working perfectly fine. We have already understood that kinase is the most common enzymatic activity in all receptors as well as free molecules in the living world. Among them, the kinase activity towards serine threonine is the most abundant. Think about any living form, think about the kinase activity. A much bigger fraction of the kinase activity belongs to the serine threonine kinase activity. What it means that both the serine and threonine can be phosphorylated by the same enzyme. They generally prefer serine or threonine for doing the phosphorylation activity. And if you think about the soluble kinase, you will again realize that this is the most abundant type of kinase. You name some of the well-known kinases which are there in the system, you will realize that they are serine threonine kinases. There are some example of tyrosine kinases as well, histidine kinases as well, but abundance or the bulk comes from the serine threonine kinase. So does this relate to the receptor activities as well? Well, probably we'll see as we move forward in the course, but like I said, that most abundant is serine threonine kinase activity. The next commonly seen activity in animal kingdom and viruses is tyrosine kinase activity, which basically means that you have a set of proteins in abundance which do the modification of serine and threonine residues. In addition, you have enzymes which have tyrosine kinase activity, which clearly suggests that they will prefer a tyrosine residue on the protein for transferring the phosphate group using that ATP. Such kinases are absent from the plant world. An example of this tyrosine kinase is Src kinase, which is found in humans as well as viruses, and they can cause tumorigenic transformation when this Src kinase is active. So when we talk about different organisms where this signaling is happening, there are different kinds of genes which give rise to these kinases. In addition to regular genes which code for functional kinases, there are pseudokinases also which have mutations which makes these proteins inactive. So in some sense, these are the inhibitory molecule because they can join hand with active molecule and become heterodimeric in the sense that one type is perfectly fine, other type is pseudokinase. So you will have heterodimeric kinase and pseudokinase combination, and such a combination is non-functional. So this is a strategy to regulate the kinase function. Then there could be enzymes or the kinase which are showing dual specificity. Dual specificity here says that they prefer or they do not distinguish between serine threonine and tyrosine kinase. They end up phosphorylating both residues. But again, remember, when such kinases come into picture, the state of the cell, the environment which is around will drive them to phosphorylate either serine threonine or tyrosine. Particularly when you are talking about these phosphorylation event, it may happen that first a phosphorylation happens at serine threonine kinase, and since the tyrosine residue is in proximity, that can also get modified or phosphorylated. Now if you think about the receptors, I spoke about different soluble kinases. If you think about the receptors, there is no difference. Over there you have the receptors which will have kinase activity towards serine threonine residue, or you can have receptors which have activity towards tyrosine residue, or you have receptors which have histidine preference. Remember, when we are discussing about receptor tyrosine kinase, when we mention receptor serine threonine kinase, it clearly says that we are talking about a receptor molecule which is largely and more often than not bound to the membrane, and the C-terminal or the carboxy terminal cytoplasmic tail, this is where you will have the kinase activity. In addition to the kinase activity, some receptors do carry tyrosine phosphatase activity, which means that you can say that do they act antagonistically to the tyrosine kinase activity? No, these are independent set of receptor which may impinge upon receptor tyrosine kinase activity, but they deal their system independently. Similarly, you have serine threonine phosphatase activity. So if you think about the diversity in terms of kinase activity which is prevalent across the world, the living world, you will realize that phosphatase and kinase activity are there integral to the receptor, and more often it is kinase activity which is integral to that, and serine threonine kinase is a preferred activity mostly in the soluble kinases, and then you also have the tyrosine kinase activity. Now let's start discussing about the receptors which you will see in different forms of life. Let's start with the simpler form of life or the microbial world. Since we can assume that life forms since we can assume that life is simpler in microbial world because they are unicellular organism, then we can say that they will probably need a simpler system for signaling. But it does not mean that since they are simpler life form, their life is any less complex or they have to face less challenges. They also face a lot of challenges. They also have to communicate to the environment. As a evolved individual, you have distinct domain of functioning or division of labor for making sure your responding system properly. But in case of unimolecular, in case of microbial world where you have unicellular structures, the microenvironment constant interaction is very important for survival. Any mistake will lead to the death. In case of microbial world, you have most abundant type of kinases are histidine kinases. I am saying that histidine kinase is most abundant type. Let me add things to there, that when we talk about the receptors, so bulk of the receptors through which microbial world is responding is histidine kinase receptor. As the name suggests, it will be targeting a histidine residue and phosphorylating that. You will also see examples of histidine kinase receptor in plant kingdom, how they respond to different cues, whether it is related to growth, whether it is related to plant hormone signaling, they utilize this histidine kinase system. But somehow these histidine kinase receptors are, or receptor histidine kinase, they are absent from animal world. They are probably there, but in the soluble form catalyzing certain reactions. The form which is present in the microbial world in terms of histidine kinase receptor, it is part of something which is called two-component system, and we will see that what exactly we mean by two-component system. So here in two-component system, we will have one component as receptor, other component as effector. Together these two combination will help ensure that microbe is responding to the environment. We will see more details of these receptors in coming classes, but you realize that when we say two component system, TCS as shown here, one component is receptor. It is also sometimes referred as sensor kinase, because this is the kinase which senses the external environment. And then you have effector molecule as a second component, which is called response regulator, in the sense that in response to sensor kinase activity, what is the signal which is put onto the sensor kinase? Response regulator will divert the signaling in given direction. One of the things which it can do is chemotaxis. Or think about a microbe which is sensitive to nitrogen levels. So all those things will be activated according to the signal, and response regulator will be different in those cases. The combination of these kinases and the response regulator, if I say RR, they work together to ensure all different responses which has to be mounted, or the communication which this microbe is establishing with its environment meets an fruitful end. One of the simplest thing you can think of to which this microbe is responding is presence of nutrient. It senses the level of nutrient and engages the combination of these two molecules through two component system, receptor histidine kinase signaling, to ensure that the cell responds accordingly. We will see in further cases that how multiple kinases will respond to one response regulator, or they will culminate into one response regulator. Similarly, there will be ideas where single kinase can eventually engage with multiple response regulator. So, many to one and one to many, both kind of signaling will be present in the system. Eventually, what they will help this organism do: chemotaxis, lifestyle change, quorum sensing, we have already discussed this, or respond to the environmental stress. Environmental stress, as I said, like suppose nitrogen availability or nutrient availability. Think about anaerobic bacteria and suddenly you have a lot of oxygen, that's an environmental stress. Temperature and so on. Chemotaxis, we understand that in search of food, sometimes they move. We had discussed in earlier classes that how cyclic AMP label in case of Dictyostelium was driving them in a given direction as a response to chemotaxis. What you wonder about is this, that what do you mean by lifestyle changes? What here I say is that some bacterium, they undergo from free living, floating and moving bacteria to something which adheres to substratum. Also, you can say when they are having food, enjoying nutrient, they are freely floating and moving around, but when it's time to come and divide, they adhere to substratum and then undergo the division. So, this is how it will be a lifestyle change which will happen over there. So, what we have understood in this class, that kinases are the most abundant type of enzymatic activity associated with receptors and proteins. Then you have different types of kinases which are active and they mediate signaling, which I mean here that whether it is histidine kinase, serine threonine kinase or tyrosine kinase. And all form of life, they prefer kinase activity receptor mediated signaling. And in particularly in case of microbial world, the two component system which is preferred for bulk of the signaling. Thank you.